So welcome, everybody. Thank you for coming to the River Center. We're really excited to share this whole series of talks. And I realized that we've been calling ourselves the voice of the river. And when we came up with the title for the speaker series, which was sort of a boring name, we decided that we would call it Nature Talks. And I don't mean that they're talks about nature, but it's nature talking to us. And I think one of the things that I really reflect on is that all things in nature have value, beauty, and fascination if you take the time to listen to them and to learn. So Westport River Watershed Alliance is a water protection, environmental group that's been around almost 50 years. I have not been here all that time. My name is Deborah Weaver, and I've been here about 10 years. And we've been in this building just about 5 years. We moved in 2 months before COVID. And it was a bad time to open a public resource building. But here you are today, joining us, as well as folks joining us on Zoom from home. So we're really happy to have you. We're also really happy to invite Dan Gibson back again because he came during COVID with a very small attendance and did a wonderful talk. And he's back here today, and he's going to give us kind of an update on the status of these wonderful and ancient creatures. A little bit about Dan. He's graduated in marine science from William and Mary. He got a PhD in biology from BU. And after that, he did doctoral research at the University of Texas Marine Biomedical Institute. And then he came to, did you return to Massachusetts? Yeah, after BU, he returned to Massachusetts and took a department position at WPI in the Worcester Polytechnic, and he advised undergraduates mostly on horseshoe crabs. He retired in 2012 and moved to Falmouth, which was a wonderful spot for him because he's right proximal to Hui, and so he can continue his research there. So a couple of housekeeping notes. First of all, if you're not a member of the Watershed Alliance, we welcome you to join us. We would really love to have your membership. We have snacks there in the front. We have restrooms upstairs on the second floor. There is an elevator if you need it. There's also stairs, and it's composting toilets, but don't be afraid, they won't eat you alive. We're really, really happy that you're all here tonight, and we're really happy to welcome Dan Gibson back again.
Thank you. All right. Well, I could talk about horseshoe crabs all day. We'll find out. I'm going to take a few of the sort of touchstone topics and go through them sort of in sort of bits and pieces, and I hope that this will be understandable. Anyway, true blue bloods first up, because everybody knows, well, they have this blue blood and it cures cancer. Well, it doesn't cure cancer, but it is used in biomedical research, and so I'm going to give that a good look before going on to some of the more fun stuff like the natural history where they lay their eggs and how to dig their eggs and all that sort of thing. Okay, so let's get these guys moving along. And start with the blue blood. This is, now can you see if I use the mouse?
Yeah.
It's not as really dramatic as I had hoped, but this guy, this poor crab, fell off a dock, cracked his shell, and bled to death. And the blood came out, and when it is in his body, just like our blood is not as red as it can be when it's circulating around, it only gets really oxygenated when it's on the pavement here like this. But what you can see is this big wad of gummy stuff, and that is a thousands, more than thousands, probably hundreds of thousands of the, we would call them white blood cells, but they don't have anything to do with immunity. They are really just plugs in the system. So these are the amoebocytes, which are so prized for the biomedical uses, and I'll get into that in just a second. So here's the sort of the long and short of it, between our blood clotting and their blood clotting. And why do we do it? Well, we have 13 clotting factors circulating in our serum, not in our blood cells. Now, platelets do contribute, but it's something that's often thought that, oh, well, we've got our clotting factors in cells. No, they're not. They're dissolved in the serum, but each one has got some little sort of protein bump on it that prevents it from becoming activated unless something comes along and knocks it aside. This little bump comes off, and then they can attach to each other, and then you get this whole clotting cascade. And one thing that's necessary for clotting in humans is calcium, and that's why they use citrated. If you put citrate in your blood bag if you donate, because that chelates all the calcium, and then you will not get a clot forming until you replace it with calcium. So more often than not, what your clotting system is doing is preventing you from bleeding to death internally, because you're springing leaks all the time, and so that is what its daily job is, unless you happen to get hurt and expose some blood to the air. And you might think, well, that's why it gouts and clots. Well, it's got nothing to do with it. The thing is collagen. Collagen is a component of every blood vessel and your skin, of course. And whenever that is exposed to these clotting materials, they get activated and they go to work. So it's exposure to the air has nothing to do with it. And you think if someone loses blood, a pool of blood sits there for quite a while. It does not clot right away, because that's not the way it works in nature. Meanwhile, their blood clot is, they have cells that carry 4 different factors, and these cells are called amoebocytes for reasons you'll see in a moment. I'll show you the movies. And they detect and immobilize gram-negative bacteria. This is an extremely non-precise reaction, but because there are so many millions of them in every milliliter of seawater, this is what they have to worry about. They get a wound, they don't want the bacteria coming in. The bacteria actually stimulates the clot that starts healing. Okay, so here is a small and transparent horseshoe crab. It's 5 millimeters long, and I'll show you the movie of his heart beating. It's just a simple tubular heart, just as the blood goes in these valves and then gets pumped out through a couple of arteries up front. But the blood is an open circulatory system. You can kind of see that the way the blood cells are just sort of drifting around in there. Blood pressure is very low, like 10 centimeters of water or something, rather than 120 millimeters of mercury. So it's very low, and they don't have a large oxygen requirement. Okay, these are the cells that are involved in the biomedical uses in the research, and these are called amoebocytes, because you'll see they move around very fluidly, and they are concentrated in the gills, but more about that in a moment. But they're fine. They carry these granules around, but in those cells, four different kinds of granules that each one has an effect on the others, and you can get a clot forming if they all go if they are all activated at the same time. So here are some amoeba sites that are not even crawling around, they're just sitting still, and they are, perfectly spherical. You might call them football sites or something. But after they have done their job, they've lost all of these granules that are inside, and they're just naked nuclei with very puffed out membranes, and they're shot. So they have to constantly manufacture more of them. So they just block wounds with them. That's what they do. And that allows for regeneration in smaller animals. They can grow back parts. The adults, it's a very, very slow process, but they do regenerate a little bit. So there's that stump. And here is one of these cells degranulating, and I'm going to show you the movie here. That each of these granules appears to pop. Now, what it's really doing is its membrane is fusing with the cell membrane. At first, just a little hole and then out. And as that happens, that membrane, of course, grows and grows because you got this big thing spreading. And meanwhile, the material that was in it is now released to the outside. And that is what makes the clot at the end. They detect endotoxins, and it's funny to say endotoxin, but endo doesn't quite get it, because it's not toxin that is in us. They're toxins that are on the surface of the bacteria. They're not in them and they're not in us. But if these get into your bloodstream, they can create septic shock and all sorts of problems. But they don't do it directly. What they do is overstimulate your own immune system, and then you get this reaction, which could cause the spiking fever, your blood vessels to dilate, so your blood pressure plummets, the blood pressure, then you get widespread clotting, and that's no good. And then finally, you go into shock, you might get sepsis, and you could die from this. So it's very important. Anytime you get an injection of a drug or anything that is taken parenterally, in other words, not through your gut, not through your enteron, that it be checked for bacterial content before it's injected. And this is a little abstruse here, but I'm just going to quickly mention what is important for the bacteria. What is the signal that the bacteria gives to the amoebocytes, the so-called amoebocytes, and what it is, they have got two cell membranes, and the outer membrane prevents a stain called Gram stain from entering. The second membrane is sort of like an insurance policy, and that means that Gram-negative bacteria never stain with this blue Gram stain because it never gets inside. So we've got two kinds of bacteria here. We've got Gram-negative and Gram-positive, to show you what those do when they have a Gram stain that penetrates. And so we have, this is crescent violet, Gram stain, and the endotoxins are on the surface of the Gram stained, I should say the Gram-negative bacteria. These toxins are part of the outer membrane, and they are what cause a huge overreaction of your own immune system. And they're called, they're lipopolysaccharides. They're partly fat and partly sugar, and they provoke this huge reaction from your white blood cells to sort of they overdo it, and then you can get any of those symptoms that I mentioned. So I want to stress there are no vaccines against these. These are very, very generalist compounds. And if you kill bacteria, let's say you have a vat of penicillin and you have some germs drop in there, and they all die, is that okay then? Is that got rid of them? No, because it's the cell walls that when they disintegrate, they actually release most of this endotoxin. So a dead bacterium is worse than a live bacterium. We have to keep them out of our systems. So in this little sample right here, these are the white blood cells, the amoebocytes, the only kind that horseshoe crabs have. And the blue blood carries, using a copper compound that carries oxygen, that nothing to do with the immune system at all. So we only get maybe 5 milliliters out of each liter of blood. Okay. So here, the freeze-dried and ruptured blood cells are put into a tube that can then be reconstituted with water that is absolutely pyrogen-free, that will not cause a fever. And then the difference is you will get, if there is pyrogen present, it will form a clot, because all of these same enzymes that were present inside the cell can still be activated, even though you've ruptured the cells and freeze-dried them and everything, they will still react. And then this is distilled water with no endotoxins in them. So as I already mentioned, every injected drug can cause a problem unless you have checked it for endotoxins. And the old test before 1975, if you wanted to test for endotoxins, you would inject the drugs into a bunny rabbit and then see if he got a fever. Well, maybe he didn't like having a thermometer up his bum. Maybe that's why he got a fever. You do not know what the symptoms are caused by. You presume that it's because you gave him some drug that you're testing or whatever, or he's got bacteria, whatever, but it's a crapshoot. So rabbit gets a fever, then you have to throw out the batch of drugs, even though it might be all right. So these cartoons were done for me by a former student of mine, and showing horseshoe crabs making their blood donations. And here is the real thing. So let's watch this for a minute. Horseshoe crabs with a needle in the hinge, and we hope missing that nerve that goes right up the center of it where the heart is. And none of these is the output from a single crab. Every time the crab stops after about a third of a cup, they take that crab out, put it back in the barrel, and bring another one on. So this facility bleeds about 750 crabs each morning, each afternoon for about 3 months of the year to get the little bit of white cell matter that's in the bottom. Now, you may have heard that it's worth $15,000 a quart. I don't know how this got into the literature, but it's foolish. And even Woods Hole Oceanographic Institution says that that's what how much it costs. But do the math. You get a quart of blood from about 12 crabs, because you have to get, as I said, a third to a quarter of a cup out of each crab. Fishermen get $2 to $4 a crab, so do the math, all right, $24, $48. Now, there is a large value added, but the companies that make this extract, they work for every penny to make this freeze-dried solution that is then so useful in testing any sort of solution that's going to be injected. And also you have to take a certain number of your batches of hypodermic needles, flush them with water that is known to be pyrogen free, and then you test that flush water, see if you've got a bad batch. Anything that is injected, not by mouth, has to be tested with limulus lysate. Now here is, this is just some news on horseshoe crab blood and how it's so useful to the effort for COVID vaccine production and so on and so on. And it's kind of a quirky little thing, but vaccines are one of the few of the injectable materials that are exempt from being tested in this way. You have to test the bottles that it goes in. You have to test what you inject it with and so on. But the manufacturing process provides the sterilization so that this is perhaps a little bit overdone. But anyway, you can see there were a lot of interest in this, particularly a couple of years ago. People were saying, alarmists were saying, we're going to run out of horseshoe crabs because they have to do so much more testing. Well, that's not the case. If those drug bottles were, if they weren't being filled with vaccine, they would be being filled with something else, and you'd still have that need, but it did not cause a great problem. But here's the color-coded reaction here, where the lipopolysaccharide that I mentioned, which is the endotoxin, if it gets next to an amoebocyte, one of the horseshoe crab's white blood cells, or if it is dissolved in the powder that is made from them, then it will activate first one of these participating enzymes, which then activates the next one, which activates the next one, which then finally causes the coagulation. And this is the flow chart. Okay, so endotoxin causes the reaction to start, and then the factor C is called, then decapitates part of the protein of factor B, and that means it's now active. It's got an active site that is exposed, which will allow it to grab onto the proclotting enzyme, which is the one that then causes the endotoxin, or causes the clot to form around the endotoxin. So that's when we have coagulagen turbidity. Now, let's watch this again. Bigger here. So you see, that's what's happening naturally, is this cell, all four of those little types of granules are rupturing and making a clot, which is then spreading out all over here. Now, you might think that you ought to be able to make the endotoxin detector without the crab. Do we really have to sacrifice so many horseshoe crabs every year? By the way, the lysate industry does kill crabs. Probably 10 to 20% of the crabs may die as a result of being bled. However, most are free. When they're set free, they recover. And meanwhile, the other use for the crabs is chopping them up for bait, to go into a bait bag and hunt for conch or eel. Now, the eels have been fished out. The conch are being fished out. So the fishermen are hoist by their own petard. They are not actually going to need that many horseshoe crabs in the future, because it's now, the fisheries people tell us that no conch, no busicon, which is a whelk, you know, it's not like one of those Hawaiian type that are so pretty, but the foot is used in squinchillo and in some Japanese dishes, and most of it is exported. No adults are being harvested anymore. And the fishermen are always lobbying for smaller and smaller size to be taken because they can't get the bigger ones. But there aren't any that are getting to sexual maturity, so I think the writing is on the wall. Okay, so how about we culture the amoeba sites? Well, it's been done. In fact, it was done in 1992 by me and my graduate student, Joan Hillig. And the procedure is pretty simple because we found that the cells proliferate in this white area of each gill flap. And now there are about 1,000 gill flaps in each one of these gill arrays, these 5 gill arrays. So we've got these little flaps here contain cells which are amoebocytes, and they contain a lot of them. Not this one is not showing the ones that I wanted, but in any case, this is where they are. You can take a few gill flaps off of the animal, culture them, rip them open, get new amoebocytes out, and then they do their thing, and then they're finished. So what did I say? How much does horseshoe crabs cost? What a fisherman gets $2 to $4 for a crab. Well, our growth serum to keep this process going cost $19 a liter. So it was not feasible, but it's because you cannot get these cells to grow in an immortal culture. So next thing though, now with recombinant DNA, it is possible to take one of the genes from the horseshoe crabs amoebocytes and use it to make a recombinant factor. Just recombinant means you do it in the lab instead of having the animal do it. So now here's a synthetic factor C, and that can, if it is stimulated with endotoxin, it will become activated, and we give it something that is not the next thing in the enzyme cascade. Instead, it's a protein that has been engineered to be broken apart by this same factor C secretion. And so at that point, it causes the molecule to break apart and fluoresce. Well, that's expensive too, I can tell you. But what I'm getting at is help is on the way with recombinant DNA. This is the first that was marketed by Lonza in Europe because they don't have any horseshoe crabs in Europe. So, hey, this is something that they can use instead. In my own opinion of it and what I have seen of how it works is it's very good for testing distilled water for purity. It does not do nearly as well when you are testing a protein-rich serum, blood serum, for instance. Does it have bacteria in it that are gram-negative bacteria? And the answer is it can't find them, it can't see them. And the other thing that is missing is you're not getting this activation upon activation. Imagine like factor C is activating factor B, in fact, the coagulation, and each one activates thousands of molecules of the next stage, So you get a bioamplification of that. And here you don't get anything. So these are kind of far off. Now, I know 3 different companies are working on a multi-factor where they got all the genes isolated so that they're able to make something that will work. And I would say the horseshoe crabs help is on the way. Oh, there we go. There's our fluorescence. I forgot. Right? on Cape Cod in Falmouth are the Associates of Cape Cod, and they have been cranking this out since 1978, I think, and that was the year that the US Pharmacopoeia approved its use as a test for endotoxins, making a lot of bunny rabbits sleep easier at night. And it has been, you use it to test all kinds of glassware and drugs and everything, as I said, that is injectable. And this is, a great company, and this is what their product looks like. It's got a little picture of a little horseshoe crab logo on every bottle. And they have just changed their logo to this abomination, which is completely indecipherable. It's supposed to be a test of these little dishes that have different solutions in them, and you can see. But it's I mean, I don't like it. I think 50 years of history. 50 years ago, they started this, and they should be proud to have partnered with the horseshoe crab. Instead, they're getting away from it because there is this idea that the blood industry is hell on horseshoe crabs, and really they're not. It's those that are taken for bait that are the bigger problem by far. Okay, so here's the problem with relying on blood that you take from a 12-year-old animal. And that is, you've got to have 12 years where nothing killed the babies as they're growing up. And we've had some rough years and we've had some good years. First three years that I researched these little guys, well, everything was fine. But then in, let's see, that was 1994, it was we had a really cold winter and several stages of cold winters. And I should just mention that the young of the year, I think I've got a picture of the salt marsh here. Well, I will have one later. That those in that situation, they don't go anywhere. They live in the salt marsh under the sand, and they burrow in about half an inch because they don't want to get down to where there's sulfide because it's black mud and they'll suffocate. So they're just stuck, between two worlds. And because the pore water never freezes, as long as when the tide is in anyway, they're okay. But imagine if you had ice forming continuously for days at a time, that crust can freeze. And in 95, we had very, very few juvenile horseshoe crabs surviving, and it's sort of like reset the clock. And from 95, that's our current crop of adults. And you got to go back 15 years. So 95 to maybe 2010, we got, finally things got restored. And now, this, by the way, is not here. This is down in Delaware. And this is when they were chopping up horseshoe crabs for fertilizer in the 30s. And this is a beach in New Jersey. And I would have said, well, I'm sorry to say we don't have this sort of thing on spawning beaches in our neck of the woods. But in fact, we do now. Let's see. Well, I thought we were. Had a little bit more to that one. No, it doesn't. It's okay. It's a good place to end. There is good news, and that was the thing that I had intended to add here, that the numbers are up. There are beaches where I have never seen crabs coming and spawning, where now there are just hundreds that you will see left by the tide, but the next tide will come and bring them in. I hope I can. This next part is more fun stuff, like out in the field and so on. And then the horseshoe crabs, green, blue, and red. We've looked at the blue for the blood, but the green eggs and the red knots and a few other things that are important. And so let's, we've seen this one already, and on to the next slide. Whoop. What I wanted to show you here is I'm a bit of an old fossil myself as well as a horseshoe crab, so I can remember when t-shirts didn't say anything, and when only sailors had tattoos, but times have changed. So this is just some t-shirts from my collection, but a third of them, I would say, and probably less than that.
That's awesome.
And then the other day when I was at the dentist, my dental tech pulled up her sleeve and showed me her horseshoe crab tattoo, and it's quite nice, you know. So I thought, well, I don't have any tattoos, but not to be outdone, I decided to get something. I went to a fair, it was a thing at the Marine Biological Lab, and they were doing face painting. You know, it was funny, a woman, so she just painted a horseshoe crab on my face, just like that. And then the next guy said, I want an electric guitar. She gets out her book. Anyway, so why do they call them horseshoe crabs? Well, it's because of this horsefoot look to it. And they're not to be confused with trilobites. Trilobites are the oldest of the arthropod groups, but they've been extinct for most of the time that horseshoe crabs have been around. So they may look like horseshoe crabs, but they're not closely related. Here are where we find them. We're 4 species. 2 are in Japan and along the Indian subcontinent and coast of China. We have the one coastal species in the United States, and it goes all the way from Maine, wherever you can find a place with no rocks, on down around to the Yucatan Peninsula. But most of the Gulf of Mexico is free of horseshoe crabs because they have this one honking big high tide every day, and then the tide goes back out, and then there's a little tide, and it confuses them when it comes to mating time. I mean, that seems to be the problem that they have, because they have to have sand. The other thing is they like to bury their eggs in sand, but if the sand is too fine, it will actually strangle the eggs. You've got to have a good oxygen content in there. So here are all of the chelicerates, spiders, ticks, mites, and so on, have got these little pinchers on the front, which are called chelicerae, and they are in place of antennae and everything but eyes. They have a lot of eyes, but they don't have any other appendages on their heads for sensory use. So they taste with these, and then they stuff food in their mouth, which is right now here, with a lot of bristles to prevent things from backing out. So, here's a crab feeding, just to show you the action there. And this is a piece of clam that's stained blue just for contrast, but he sucks that in, has no teeth of any sort, but the spines bring it in. And if you give a large crab a razor clam, you just suck that thing and just breaks the shell away and just sucks it up in a very short time. Okay, so these are some of the other of the chelicerae group, and in fact, these horseshoe crabs have just been recatalogued to be right in the middle of the spiders, scorpions, and all of that ilk. Because look at it, here's the horseshoe crab without his shell, without his top shell. Does that look like a spider to you? No, yes, it does. Okay. So when you find one on the beach, and you may have seen these little signs that say just flip them if you've ever been down in New Jersey, that's all really that they require. They can use that tail to flip over underwater, but they're too heavy out of the water. So when the tide comes in and they get beached, then if they're on their backs, the gulls may come and pick their gills out because that's the softest part, and that could be, that's terminal. But if you turn them over, they will find the water again. But why are they out of the water? That's the big question. Let's go on here to see. So this is down in New Jersey, and it's a site along the shoreline when the tide is going out. Am I reverberating?
Okay, not bad.
Right. Well, so here these guys are all sitting. I can guarantee there's a female at the center of every one of those humps. But meanwhile, let's just go back and look at this again, I think. Meanwhile, all of these others that are up higher on the beach, the tide was in there. Everybody says, oh, they come out of the water to spawn. They do not. They come out to where they're just like got the tide on their foreheads and they dig their nest, or they may not even come that far out. But those that venture up as far as they can go will sometimes get left by the tide. Something like 1 million horseshoe crabs per I can't remember how many yards it is, will die down in the New Jersey on any given spawning day. So that's their.
Can they survive the turn of the tide?
Yes. Well, what they do is as long as they burrow in, keep their gills wet, they're fine. So here's one who's just headed back for the water. And now here's the other part of the equation. As you might have noticed, there are a lot of eggs just swirling around in the water near shore. That's what the birds eat. The birds, with the exception of the godwit, it's the only bird that's got a bill long enough to dig down to where the eggs are laid, because the crabs will come up and dig a nest that is maybe 8-10 inches down, and that means it has to be a well aerated beach. If it's waterlogged and sulfurous, they won't do it there. But these red knots are cousins of robins, and there's the flock right now that comes through Cape May, New Jersey, is only about 23,000, but it used to be 10 times that big. And they come and they time it so that they land when the horseshoe crabs are laying eggs. And if they don't get a good meal of horseshoe crab eggs, then they may stay there for a week and really chow down on them. They will not make it to the Arctic to spawn. They fly from, believe it or not, Tierra del Fuego, that's their summer grounds where they grub little clams and snails out of the mud down there. And then the red knots, and so not the horseshoe crabs. And then they fly all the way They make a stop in Brazil, maybe one somewhere on the Mexican peninsula, Cape May, New Jersey. That's where they get their sustenance to fly onto the Arctic. And Louise Russell, late Louise Russell, who was an artist on the Cape, did this wonderful 7-foot tall map of these, and this shows their return flight. They don't really turn black, but it's just to show how they do. And we get more of them in the fall, but then if they come to our beaches, they're eating little baby crabs instead. So we have a threesome there, and only what? I never plugged in. So I'll stop it over here. I can, but I'm gonna have to.
Power strip right
Oh,
behind on the right
yeah.
behind it.
Yeah.
Dan, we're just about out of time. Want me to plug that in for you? So we're just about out of time. for our presentation.
All right, let's see if I
Yeah.
can see.
Yeah, good.
Yeah.
All right.
Well, while we're waiting for Dan to finish up his presentation, I just wanted to alert you to our next presentation. Every last Thursday of the month, we'll be doing something. So February 27th is Curie and Finley from the Westport Sea Farms talking about oyster farming and that industry here in Westport. March 27th, Annie DeMonte is talking about whaling and whale preservation and protection. And April 24th, we're doing Charles Clarkson, who's doing something on the rising tides and how that's impacting the habitat for salt marsh sparrows. So these are all the last Thursday of the month. And as I said earlier, Paul Dobbins will be doing our annual meeting on March 9th. Okay.
All right, so let me rapidly tell you why these crabs bother to come to the sandy beaches, and that is oxygen. If any of those eggs that are laid below the tide line, where the tide never goes out, they will never get oxygen when the pore water drains out at low tide, and they will strangle and die. So this is essentially, see, this is a difference. You can't tell when you're looking, but the crabs can, and they have oxygen sensors in their gills. So here's and you see the bubbles that are rising from this crab are actually from the sand. It's aerated sand, and that's how she knows it's time to lay a nest. And if you don't see the bubbles as you're watching a pair of them, they're not mating, they're just hanging out. How many eggs? Maybe 5,000, 10,000. So there's, yeah, per lay. And there's where they make these nests. And when the tide is out, you might find something just like this.
Yeah, we'll try and
Okay, now someone was. You're gonna cut me off.
Yep, wind it down. I'm sorry to say.
Okay. Well, this is my fault to get sort of behind on where my things go. But did you see? That's how you find the eggs.
Yeah.
Okay? So
Wow.
2 to 4 weeks if the sand is warm. And they wait to the next high tide, spring tide brings them out. So, okay, well, I've got more material, so I hope you ask me back. for-
Yes, good. So let me just check in with Tim over there, if anyone's given us any questions in the chat online.
We do. So one of the questions we have is, how many times do they molt in a lifetime?
It takes them somewhere between 11 and 13 molts to get up to adult size. And I suspect that some years, when it's a kind of lean year, some of them will say, all right, I'm going to become an adult now, and I'm going to do it at age 9. And you'll find little males like this, and sometimes very small females. So that's a sort of a short cycle.
Another question we have is, do you have any thoughts on how the industrialization of the continental shelf is affecting their numbers? Does it impact their migratory routes?
It can, but it's a worry for the birds. The horseshoe crabs will find a beach. They will, if they can't find the one that they went to last year, they aren't salmon. They will not go back to the same stream necessarily. They're separate populations, but that's not a big problem for them. For the birds, it is.
Thank you. Another question we have, they're wondering if you can talk a little bit about local conservation efforts. Maybe you could touch on the new regulations for fishermen.
Okay, fishermen have used to be able to take 1,000 crabs every day of the spawning season, every damn day. And now it's down to 400, and it's only on the days that do not surround the full or new moon. Now, that's good. It doesn't necessarily solve the problem, because they're not linked to the moon tides the way you might think. They don't need to go up to the highest high reaches to lay their eggs. But in general, we've added 20 additional days in the spring for spawning where they will not be bothered. Crabs are coming back. So there's that. The crabs are coming back.
That's good to hear. And I'll have one more online question for now, and then you can ask the audience. That lines up exactly with the next question. And someone was wondering, how closely does their appearance line up with the high tides? Can you see them before or after?
Well, as it happens, if you get there right at high tide, which is what everybody does if they're doing a survey to find out how many crabs there are, they may be there, they have been and gone, there's a really high tide, or they may not be there yet. So this can give you kind of a false positive or a false negative. So they have to have a beach. They are intertidal. They lay their eggs intertidally. The tide has to sweep over this and wet it down, and they lay their eggs. And then when the tide goes out, they get aerated twice a day as the poor water drains out.
Okay, we have one more question that was right in the back, and then
I
we'll.
just want to clarify one thing about regulations. There's now a closure as of last year, from April 15th to June 7th.
Derek,
Yeah.
hi.
Okay.
Yes, okay, I stand corrected. I should have remembered that from things that we had done.
So thank you all for coming. Really appreciate having you here tonight. And Dan is going to be here for a few more minutes if you want to come up and just talk to him one-to-one.
Love to.
Welcome you to do that. And we really appreciate having you all here, and I hope you'll come back and hear the rest of our nature talks.
Thanks.
Thank you.